We've been focusing on the feature extraction properties of individual cells in V1. Now let's turn our attention to the overall structure of V1. And that structure can be illustrated abstractly using what's sometimes referred to as the ice cube model. So in the ice cube model here, different cell types of the visual cortex are organized into orientation columns. Now, cells within an orientation column, they share the same rough receptive field position and orientation. And each column has cells of each of the types that we've discussed and of each of the different representations. So here we've got our orientation columns. Those orientation columns are grouped together within the visual cortex into ocular dominance columns. And each ocular dominance column contains cells for all the orientations that show a preferential response to the same receptive field. So here we have two ocular dominance columns. Ocular dominance columns are then organized into hypercolumns, consisting of an ocular dominance column for each eye, corresponding to the same receptive field on the retina, and containing cells from each orientation. And so that combination of two ocular dominance columns creates a hypercolumn here. Finally, within those hypercolumns, we see cortical pegs. And cortical pegs are regions where one finds cells specific for colors. Pegs, also known as blobs, don't appear in area or layer 4 of the visual cortex, however. So here we've got our pegs. Now that abstract simplified map is quite different from the sort of picture that we get with imaging. So when we look at this image here, which is an image of area V1 in a cat, what we see is we see a map of iso-orientation preference contours along the black line. So different orientation preferences, they have areas that are spinning out around this central pinwheel structure here. Ocular dominance boundaries are then indicated by these white lines. And the spatial frequencies are indicated by color coding. So red regions correspond to low spatial frequency preference and violet to high, and they go from red to violet. Here's a different imaging technique used to spell out those same sorts of structures. In this case, though, we're looking at the functional organization of the visual cortex of the prosimian bush baby. And our technique here is optical imaging of intrinsic signals. So what does all that mean? Well, first let's just show, again, we have this pinwheel organization to the cortical structure. Different orientation columns spinning out from this central nexus. And here we see the development of ocular dominance columns in an animal. And we're looking at time steps T0, 10, 20, 30, 40, and 80. Each pixel is going to represent a single cortical cell. The colors represent ocular dominance of each cell. Red indicates complete dominance by the right eye, and green indicates complete dominance by the left eye. Blue represents equality of the two eyes. And so we can see here, starting off at T0, there's very little dominance in the representation in our V1 area. In 10, we start to get some. 20, it becomes stronger. 30, until by the time we get to 80, we have these well-defined ocular dominance areas. Now, a lot of these images of the visual cortex of animals were taken using this optical imaging apparatus, right? So these are two researchers. Zhu and Casagrande, and they're standing in front of this apparatus that's used to do this optical imaging technique. And then what happens is they shine colored light down, and it detects different red light reflective properties of dioxyhemoglobin, so two oxygens bound to a hemoglobin, versus oxyhemoglobin. So again, it's a metabolic measure, this time using light to detect the difference in the metabolic use of oxygen by different areas of the brain. Even with all that, we can still get a sort of slightly more realistic yet stylized ice cube representation of the overall functional architecture of V1 here, where we have particular orientation columns arranged in pinwheel fashion, ocular dominance columns sweeping across them, blobs in between these areas, and so on. Similarly, we see the retinotopic mapping of V1 in this pretty old, actually, from 1988 image. So what they did is they flickered this image in front of a macaque. And then they used a Cytox stain to indicate what areas of the layer 4 V1 in the macaque were excited by this stimuli that they flickered on and off. Right? And so what they got was this representation of this pattern in the cells of 4C and V1 using the Cytox staining. So the lateral geniculate nucleus, as well as V1 in our visual cortex, have this retinotopic mapping. It's not a one-for-one mapping, all right, of the visual field, because the resolution for the foveal area is much, much better than the resolution as you move away from the foveal area. And so here are two different ways of getting at that point. So they imagine if you unfold the striate cortex, it's going to have a shape, they say, like a pear here. And you're looking at the relative distance from the foveal cone peak, the center of the fovea, and you're seeing how much of that cortex is used to represent 5 degrees, and then 10 degrees, and then 30 degrees, and so on. And as you move farther and farther away from that foveal cone peak, we have smaller and smaller bits of our visual cortex being used to represent that area, so the resolution gets worse and worse. And this similarly, we have this picture of a person here. If we were to look at that person at the back of our eye, of course, because our lens inverts the image, that person would be upside down. But when we get to the cortical representation of that image that's collected at the eye, what we find is that the resolution around the foveal area, right here in the center, you're looking at her nose, right? That's very high. And the farther we move out from that foveal area, the lower the resolution gets. Now, topographic maps are actually a common coding technique or strategy within our brain. So we can observe in both the motor cortex and the sensory cortex these homunculi, these topographic maps. And in this case, they're also distorted. So in both the sensory and the motor cortex, what we find are the areas of the body where we need the finest representation, the highest resolution. The hands and the face primarily are the largest areas of those cortex relative to the size of the body part that they represent. Finally, we should note that if we have damage to our striate cortex, that causes blind spots in the contralateral visual field. So if you damage the right-hand side of your visual cortex, that damage will result in a contralateral visual field blind spot. And it's called cortical visual impairment. The exact nature of the damage is going to be determined by the specific nature of the impairments, of course, but if I were to damage an area of your visual cortex, it might result in you getting this sort of strange blind spot. You can't see things in that particular area.